Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, a part of the Finding Genius Foundation. I have Barak Rotblat. He's a senior lecturer at Ben-Gurion University of the Negev, as in the Negev Desert in Israel. We're going to talk about a new class of RNA molecules, long noncoding RNA. And Barak runs the Rotblat lab. So, Barak, thanks for coming. How you doing?
I'm doing well. Thank you, Richard. It's bad.
Thank you for being here. Tell me about your research.
So I can tell you how I came about to do it, and maybe it'll give a bit of a flavor to it. And what I'm actually postdoc in Vancouver. It was around 2010, and the people were starting to discover these RNA species, which keep coming up in experiments based on next generation sequencing, and which do not code for proteins. And people knew that there are some RNAs that don't code for proteins, but the thing was that there were so many of them, so many different RNAs that just don't code for proteins. And at that point, I was thinking that if there's so many of them and they're changing according to different biological and so on and so forth, they must be doing something interesting. And I thought it would be very exciting to go and study what these RNAs are doing.
How many are known? How many non-coding RNAs are known about?
So people in the field can have some debates about this, but I think we can safely say now that we know of about 100,000.
Oh, wow. How does this compare to DNA? I know we have exons, which contain the coding portions, and then we have, I think it's called the introns, I'm not sure. But the non-coding portion of our DNA is pretty extensive. How does that compare to the amount of non-coding RNA that's been discovered?
So that's also a thing, because these kind of discoveries, they let us start thinking about how we define a gene and how we treat the genome, the sequences embedded within it. So if let's say 2% of the gene, it depends how you define it, but let's say 2%, 10%, maximum 20% of the genome, the sequences within it eventually turn into RNA, which eventually turns into a protein. Now, you can have a very large DNA sequence, which the RNA coming from it will actually be quite small because of a process called splicing. So this piece of DNA is being transcribed or copied into RNA. And then there are a bunch of proteins who bind this RNA and manipulate it in ways which remove some sequences and stitch it, like cut and paste mechanisms, which remove sequences and stitches back to the rest. These are called exons. They leave the nucleus and eventually are translated into protein. So if you think that we have about 20,000 genes in the genome, which is about 2% of the genome, which their products are actually these RNAs which turn into proteins, then you can think about the rest of the genome, or maybe 90% of the genome, which is described into RNA, and this RNA is being spliced just as if it's a protein code, but actually they don't code into proteins, or maybe, and that's what we think.
So there's no one-to-one correlation of the amount of non-coding DNA and the amount of non-coding RNA.
No, it's not one-to-one correlation. And now we know that these long non-coding RNAs, they are genes just as ones which are eventually translated into proteins. So structures, they have sequences which tell them when to turn on or off. These are called promoters and enhancers. And then they go all this processing, which we discussed, which is splicing and other RNA processes. So these long non-coding RNAs, they actually look like a normal gene and they behave like a normal gene, probably going through Darwin selection like a normal gene, except that their function is defined in the RNA molecule itself rather than the protein which is being translated from the RNA.
Where do these RNAs come from? Do you think they could be trafficked through extracellular vesicles from other cells? Or do you think that, if you have a virus that enters a cell, an RNA virus, what's the difference between that and a long piece of RNA that's in the cell? Like, where do all the these long non-coding RNAs come from, do you think?
So first of all, they're encoded in our genome. So there are promoters, which are sequences within our genome, which binds proteins, which actually synthesize the RNA. And these promoters tell genes, also proteins, when to turn on or off. So these long non-coding RNAs, they basically function the same. So they're not necessarily uptaken. A cell can uptake nothing, and still it will make all these long non-coding RNAs. And what is interesting, there are two interesting features in these long RNAs when you compare them to let's say the canonical regular RNAs, which we used to. One feature is that they're not very much conserved in evolution. So if you look at let's say a worm, right, like C. elegans, will have 20,000 genes encoding to proteins. We as humans have 20,000 encoding to proteins, and many of the genes found in worms, there is something quite similar in the human genome. However, when you look at long non-coding RNA, then C. elegans has a few hundreds, maybe a few thousand. But in our genome, there are 100,000, or at least tens of thousands of genes whose products are long non-coding RNA. So the repertoire of genes whose products are non-coding RNA has expanded through evolution very rapidly as compared to protein coding genes. So this is one feature which makes them quite interesting. And if we take another example, a mouse, which is another model organism, which is a mammal like us, if you look at the genome of a mouse and a human, it's very similar. So if you look at the protein coding genes, we're 90% identical. But if you look at the long non-coding RNA, humans and also primates, we have 2 times more genes who code for long non-coding RNAs as compared to mouse. So demonstrating again the expansion in evolution of this class of genes, which I think is an interesting angle to think of how nature has increased complexity very quickly. So I think it's perhaps by expanding the number of these genes. And the question is, how a long non-coding RNA gene is born? It's a question which people are studying. And I think one of the leading ideas in the field is that there are transposons. So these are sequences in our genome which can jump around. And they will jump somewhere and bring a promoter with them, which means that this piece of DNA, perhaps it was a gene desert, now is being transcribed into RNA. And if this RNA happens to acquire function, then it will be maintained by evolution and so on and so forth. So this is one of the ideas.
What do you think the function of these long non-coding RNAs is?
From the tens of thousands of long non-coding RNA, and there are only, let's say, a couple of hundred who have been rigorously studied functionally. And these studies are not so simple for 2 reasons. So one reason is that if we let's say, I find a interesting gene in cancer, which is the system we're working on, and it's a protein coding gene. I can look at the sequence, and from the sequence, I can know what is the sequence of the protein which encodes. And then I can look at other proteins which are similar to it in sequence and most likely in structure, what they're doing. And it can give me a good edge to start studying this new gene. So, for example, if this new gene will give product to a metabolic enzyme, and it's very similar to a metabolic enzyme that exists in yeast, and we know that in yeast, this thing breaks down this and that compound, then I can start by the study of this new gene by asking, well, perhaps it's doing something similar. Now, with the long non-coding RNA, because by definition they do not code for proteins, then when we look at the sequence of the long non-coding RNA, there's very little information regarding what it might do. So one way to go about studying long non-coding RNAs is to start from a disease. So we're, for example, studying cancer. And if we find the gene, which is, for example, very active in tumors and not so much in normal tissue, then we can start asking, okay, perhaps this gene, which in our case would be a long RNA, perhaps this long non-coding RNA has a function in this system. So we do what we do in the lab, which is to manipulate the genome of tumor cells and other cells, and, for example, remove this long non-coding RNA gene and then ask, so what is happening to this cell? If it's a cancer cell line, then perhaps we would go and asking things which are related to cancer. So does this cell proliferate more, grow more? Does it migrate more? Does it invade more? Is it now more or less resistant to chemotherapy? And this is the kind of directions we like to take, because I think it would be very challenging to just pick a random long encoding RNA and try to study it without having like some kind of biological hook about what it's doing.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now, back to the show.
Well, is the name even accurate? Do we know that these non-coding RNAs if they're non-coding, what do they do? And the ones that have been studied, what's been learned about them? What kind of things do they do?
Regarding long non-coding RNA, so we define them by what they're not, right? The definition is by what they're not. This is always a funny definition. So they're not coding for proteins, okay? But do they not code for proteins? And the idea that they do not code for proteins comes from several observations. So one observation is that if you take a random piece of RNA and it is long enough, it has, let's say, a few thousand base pairs, just by chance, you will have an ATG come along, like these 3 base pairs, which symbolize the start of a translated protein. And then you'll have 3 more, the next 3 and the next 3 and the next 3 will be also different base pairs, which eventually represent some kind of amino acids, which are synthesized into a protein. And then you'll have a stop. This I'm saying just by chance, if you take A, C, T, and G, or U, in the case of RNA, and you mix them up and you make a sequence, just by chance, you'll get what is called an open reading frame. So the sequences that basically define the genetic code of the protein. And if you have an RNA which has a coding sequence on it, and it's longer than what you expect to find by chance, then we defined it as a long encoding RNA. So who said that they're not small sequences or not small proteins encoded in long RNAs? Well, apparently there are, and we'll get to that in a minute. So this is one observation. The second observation was that when people did studies such as polysome profile, where you ask, doing translation, where does the ribosome sit on the RNA? And basically what you find is that there are a lot of ribosomes sitting on the region of the RNA that has a coding sequence. And then the ribosomes drop off the RNA when they reach the stop codon. So if you do this kind of ribosome profiling, you can actually map what is being translated on an RNA. Now, when you look at the normal protein coding RNA, you see that the ribosomes, there's a peak of ribosomes at the start, and then it occupies the sequence, and then the peak drops at the stop. So in long non-coding RNA, often you find that this is not the case. So the ribosomes seem to be scattered randomly on the RNA, and this second observation is also help push the idea that these long non-coding RNAs do not code for proteins.
It's kind of like junk DNA. I mean, why would there be so much of it if it does nothing? It makes no sense. But go ahead.
Yeah, it's perfect comment because we're an undergrad and I was studying biology, and they told us about junk DNA, and basically referring to there were some ideas like it's a backup for evolution. There's a lower chance that actually the damage will hit an important spot and so on, what this junk is doing. But actually this junk is being transcribed to RNA, and many of these RNAs are long encoding RNAs which are active. Now, the reason why when I was an undergrad we thought this junk DNA is not being transcribed is because of the technology. So the technology before, if you wanted to ask what RNA is in a cell, you'd have to make a bunch of probes and use these probes to fish out the RNAs and ask, okay, throw them out as fish hooks and see if you catch any fish. If any RNA sticks to these probes, which will basically report that this RNA exists in the cell. So you had to ask for. Now, in the new sequencing technology, I'm calling it new, it's not so new anymore, but still, what we do is we take all the RNA and we stick it in a machine and we get the results in the email, which basically unbiasedly all the RNA that there is in the tissue or in the sample that we put in the machine. We started doing these kind of experiments and not asking, is my favorite RNA there, but rather asking what RNA is there. That is where we found all these RNAs.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And basically what they do, at least from what we know now, is the regulators. So they have regulatory functions. I would say the regulatory function is, of course, a very naive way to describe what a gene is doing, but let's say we can define some genes, they have structural functions, for example, the one that build the cytoskeleton. Some will have metabolic functions, for example, an enzyme that breaks them. And some are regulators, which basically means they're genes whose protein products, for example, they would bind one protein, an enzyme, and turn it on or off, or they would bind the protein and modify it, and then it will form the scaffold and so on and so forth. So what these RNAs are doing, they're binding the genome, so they're binding our DNA, they're binding other RNAs, and they're binding proteins and regulating their activity. So for example, they can bind a promoter region of a gene and turn it on or off. Or for example, they can bind one protein with one region of the RNA and another protein with another region of the RNA and form a complex. I mean, the classical RNA protein complex is the ribosome, of course, but now we know that there are many complexes in the cell which are very dynamic, not so stable like the ribosome, and they're regulated by RNA. So these long encoding RNAs, they're actually quite fascinating in the sense that they're having so many regulatory functions. And if we think about it in a more theoretical or philosophical way, we can think that the expansion of these long encoding RNAs in evolution is supporting the increase in complexity, because basically increasing biological complexity is very much achieved by having a more and more sophisticated regulation, especially over gene expression.
How would you compare the regulatory ability of these long non-coding RNAs versus epigenetic marks versus methylation, histone modification, etc.?
It's a great question because we know, for example, what enzymes they methylate CpGI, right? If you take this enzyme, if you purify it in a tube and you add a piece of DNA and it has a CpG on it, it will methylate it, right? And this usually is epigenetic marks of gene silencing. We have histone modifications. So the enzymes, if you purify the enzyme in a tube and you put histones in there, it will methylate it or acetylate it or do many other types of modifications you can get. But the question that we're still trying to understand and figure out is how these enzymes know where to go in the genome and do their job. So how does the enzyme which methylates a particular histone of a particular gene know to methylate that histone of that gene? Because presumably it's floating around in the nucleus and it's emitting many histones. And how does it know where to go? And this biological question is fascinating. Of course, it's a very vivid and exciting field, the epigenetics and gene regulation, gene expression regulation. Very, very exciting field, and people are doing great work. And one of the ideas in this field is that perhaps these long encoding RNAs, this is their function. So you can imagine a long encoding RNA, which on the one side of it grabs an enzyme which can modify a histone. On the other end of it, by complementation, it binds to a specific region of the genome, and there you go, and you have the enzyme brought to a specific region of the genome. Now that I think of it, it's very much like how CRISPR works. But this is one of the ideas of how these long encoding RNAs are actually linked to epigenetic regulation.
So are they the architects of epigenetic marks? Do they facilitate the adding or removal of marks? Is that what you're saying?
That's what I'm saying. And this is a very general saying, and in biology, of course, you always have to be careful because we know only what we know. But I think there are evidence, there are long encoding RNAs have been thoroughly studied and have been shown to bind proteins which modify histones, for example, and bring them to specific regions of the genome where they do their job. Another interesting feature of this long encoding RNA is their location within the genome and how they function in cis. So you can imagine a region of the genome where RNA is being transcribed from it. This changes the biophysical property of that region of the genome. There's a lot of discussion about phase separation, and I don't know if this is exactly the same thing, but you can imagine that there's now a molecule of RNA in a region of the genome where there was not. And if you have proteins which have affinity to RNA, their effective concentration in that region of the genome, and then just by being transcribed, these RNAs have been shown to regulate their neighbors, basically the expression of their gene neighbors. And one very nice and paper that was published, and what they showed was that there's this gene very important in heart development in mouse, and called HAND2, and it has a neighbor, which is a long non-coding RNA called upper hand. And what they did was that if they deleted upper hand and they stopped its transcription, basically, then the next door neighbor didn't function, and there was a defect in the development of the heart in mice. But if they swapped the sequence from the long non-coding RNA sequence that was there to another sequence, in this case, it was a protein coding sequence, it was still functional. So in this case, we have a long non-coding RNA that is transcribed from a specific region of the genome whose job is to regulate the neighbor just by being transcribed, and is duly independent of its sequence. And this is, again, adds the challenge of studying these RNAs because there's not much functional information embedded in their sequences.
How can you tell that there's a certain number of different kinds? Like, what does the processing look like to discover, the isolation, the separation, and the processing of a sample to find these non-coding RNAs? How do you know that you've got one, but it's not chopped up fragments from other pieces of genetic material?
So the way we do it, there are several ways. So in this regard, it won't be different than studying any other kind of RNA, let's say protein coding RNA. There too, you're always, when you do experiments like real-time PCR, experiments which allow you basically to quantify how much of this specific RNA, you always have this challenge of saying, how do I know that I don't have contamination? So there's several ways to control, and one of the popular ways is to just extract total RNA from the cell or from your tissue, for example, with trisol, and then you treat the sample with DNAs. So it's an enzyme which chops up DNA. And then you remove the DNA, and you continue on with the processing of the RNA. So even if you have some leftover DNA, you just get rid of it. So it's this, I don't think, is a big challenge in the field. What is an interesting challenge is that the localization of these long non-coding RNA is very much important for their function. So, for example, there are long non-coding who function in the nucleus and they bind to chromatin. So you can, and people have done this, they isolated chromatin and used RNA sequencing to see which RNAs are bound to the chromatin. So they found some long non-coding RNAs, and later they discovered that these long non-coding RNA have function in binding the genome. For example, regulating the epigenetic landscape. There's some long non-coding RNAs who bind to proteins and RNA in the cytosol and regulate function. So and people have done this too, they isolate cytosolic RNA and dispose of the nuclear and the mitochondrial and membrane-bound RNAs, and then they sequence it and they see what's there. So they find some long non-coding RNAs which are there, and then you can ask the question, what are they doing there? Who are they binding? And so forth.
A lot of different possibilities.
Yes.
What is your research to just slowly attempt to characterize the function of the rest of the thousands of long non-coding RNAs, or like what's your focus right now?
So basically what we do is we think about some ideas which we're interested in, for example, long non-coding RNA, and then we look for clinical relevance because we study cancer biology. This is also our biological model. And so we look at cancer data where people have taken tumors and sequenced the gene expression or sequence of the RNAs there. And we look to see if there's any indication that the gene we're interested in, or we rank the genes which are, let's say, the most interesting to the least interesting based on their clinical characteristic. For example, we worked on one long non-coding RNA, which is called TP73 antisense 1. And from here forth, I'll call it just the antisense. So we saw that the reason why we were interested in this gene is because it's a neighbor of a transcription factor called P73, a member of the P53 family. So when I was doing my postdoc in Jerry Melino's lab in the MRC toxicology unit in Leicester, I said to Jerry, look, there's a long non-coding RNA next to our favorite transcription factor. And we got really excited to see if it might be regulating its neighbor. It was very early days of long non-coding RNA. And we did some basic experiments and they looked very promising. So then we went to the clinical data. And what we found was that it is really highly expressed in brain tumors versus normal brain. And this is already an indication. Now, in this case, we don't know if the gene does anything, but there is a chance that it's important in this disease. What we also found is that if you take patients whose RNA was profiled in tumors and you divide them into two groups, one which have high activity of this gene and one which have low activity of the gene, and you draw their survival, you see that the group that has high activity of this gene, they survive less. So taking these two indications, we said, okay, it's clinically relevant, so it might be doing something important. And then we went ahead to study it by doing basically gene manipulation, knocking it down and seeing what happens to the cell. And my first student in the lab, that was her project, and she was working on what it does in a specific type of brain tumor cells, which are called cancer stem cells. And it was very disappointing because basically it did nothing. And we thought this is okay. I mean, some things are expressed in cancer but do nothing. This can happen. But then she asked to do one more experiment and to try to see how they behave under chemotherapy, because she said all the patients with this kind of brain cancer, it's called glioblastoma, they go through the same chemotherapy. And if this gene were to be involved in the response of the tumor to chemo, then it can explain why patients with high expression, they have poor survival. Perhaps they are more, the tumor is more resistant to the chemo. So she tried the experiment and it worked. So this is the function of this gene. And we tried to understand how it's doing so. And we saw that it regulates the expression of many, many genes, many metabolic genes. But one of them is, it's an aldehyde dehydrogenase. So this gene is basically, the product of this gene is a protein which breaks down alcohol in our liver. And when she gave, so there's this drug they give to alcoholics, which blocks this enzyme. So they take one shot and they get an instant hangover. And she gave this to the cells and they lost the protection. So basically we think that the function of this gene is to regulate the expression of detox enzymes. And this is why the brain tumor cells, they like to have a lot of it.
Is your goal, again, to catalog the function of all the non-coding RNAs out there? Or what are your specific goals for your lab?
I don't think I'm not really into this doing the very high throughput. I just want to more cherry pick something which is interesting and drill down and try to understand it in a deep way. So,
quick question: are you guys looking to see if these long non-coding RNAs are ending up being packaged inside of extracellular vesicles? Is there any investigation into that and characterization now that we know that they exist? Do they show up as cargo in these things?
Yeah, they show up as cargo, and it has been shown that they can be uptaken by cells and be functional in the cells which uptook them. Yes, this has been shown. This is not our line of investigation, but yes, it's known.
Well, it's just interesting. It gives if you're studying particular ones that you find interesting, you may also want to look into see if those specific ones you're interested in end up being packaged into these exosomes or extracellular vesicles. And if so, do they serve the same function in the cell that creates them as in the recipient cell that takes them in? Just another nuance to their action, possibly.
Yeah, that would be really interesting. And now that I think of it, it can be interesting also in the sense of the tumor microenvironment, because of course you have a lot of communications between the tumor cells and the normal tissue around it, or it's not really normal anymore. The tumor cells they change it, and epithelial cells and immune cells which come there, and it can be interesting to see what, for example, an immune cell which presumably might uptake this long encoding RNA, how will it react perhaps to chemo? That's interesting. I don't know if they're being excreted from the cells, but it will be interesting to check.
You said earlier that these non-coding RNA will literally have multiple ribosomes bonded to them, like an entire ribosome?
Sometimes they will have, sometimes they will have many ribosomes attached to them, yes. So, and the funny thing is that they have many ribosomes attached to them, but because of the profile, that it doesn't go up near the ATG code and down near the stop codon, then people have assumed that they're not being translated. But there's a paper that came out, I think a couple of months ago, from the group of Jonathan Weitzman, and they asked this question. So are these long non-coding RNAs really non-coding? And they did a very thorough investigation of ribosome profiling, and they actually found short protein coding sequences within some, I would say, I don't remember the numbers, but maybe, 5%, but I'm not sure. Some of these long non-coding RNAs. And actually they collaborated with another group, I think in Max Planck of Matthias Mann, and they found by mass spec over tissue, they found these small proteins actually in the tissue. So these RNAs have the profile as if they're being translated, and there's peptides or proteins which are corresponding to these sequences. And then they went ahead and they confirmed, I think, 2 of them, which they showed that they're actually biological function. They have a biological function. So when I saw this paper, I said to my student, her name is Gal, why don't you look in their data and see if our long non-coding RNA is there? And it is. So now we're very excited to see if this small protein, which is encoded in our long non-coding RNA, is functional, and perhaps it also plays a role in what this gene is doing. So this is a new project in the lab, and we're excited by it. And another interesting aspect of this long non-coding RNA is that it is conserved in monkeys, but only old world monkeys. So new world monkeys and let's say down in evolution, it does not exist. So it can be interesting if it has a function in the brain, which this gene is usually more highly expressed than other organs. Then maybe this small protein is involved in our brain. But this is speculation.
Yeah, I'm just curious about viruses. If you have an RNA virus, and I don't think you'd have the answer to this question, but for people that do work on viruses, it'd be interesting. Mm-hmm. What if a long string from a virus enters a cell and then ribosomes attached to it? I wonder if it acts at all like a non-coding RNA, and if this just sheds more light on what could happen to, for instance, viral material that enters a cell. From what you study, I just wonder if there's any corollaries and any other additional knowledge to be gained.
So I think a lot of these technologies and these biological principles, they actually come from viruses, because the virus will sort of, in very simplistic terms, inject its RNA into our cells. And actually our ribosomes will bind this RNA and turn it into a protein. And this is how our cells make more virus. And what is interesting in virus...
I remember you spoke about the patterning of how the ribosomes will bind to a particular piece of RNA. And you mentioned with the non-coding ones that it appears that there's random binding, but with other RNAs, it appears to be deliberate, structured binding. So I wonder if people could evaluate, again, viral RNA that enters a cell and see if there's structured or unstructured binding of ribosomes.
So in viruses, we know what it is, the type of binding, let's say, and it's very structural. Actually, it's the viruses, they're so good at exploiting our own ribosomes. I think sometimes they're more efficiently translated than our own genes. So this actually, this has been figured out. Yes, and what is also interesting is that they can have multiple proteins coming from the same RNA sequence. And this is also very exciting, because you can imagine that there's an ATG and then three codons and the next codon, which is 3 bases and 3 bases and 3 bases, and this will make the protein. Now imagine at some point there's another ATG, which is frame shift. So it's not the 3 base, it's not going by 3 with the first ATG. And then it has the other part of the sequence will be a totally different protein. So viruses exploit a few open reading frames in one RNA to make a few proteins, and they're super efficient. Actually, we don't do that. Our own genome, our own RNA does not do that, but viruses, they're like the acrobats of RNA and how to exploit our own system.
Yeah, that's amazing. So people have done profiling of, again, viruses once they're inside a cell, and they look at the arrangement or placement of the ribosomes, and does that give them any clue as to, I don't know, how our own RNA, what makes an RNA acrobatic, as you said, or efficient or non-efficient? Like what, I don't know, does this give you any insight into, depending on the sequence of a piece of RNA, the functionality comes from there? Is there an underlying language or code that can be figured out based on, the binding of the ribosomes, where they bind, how they bind, et cetera? Do they bind and then come off and then rebind in different spots? I mean, has any of that been observed?
Yes, so this has been observed. Usually what they do is they bind and then something happens, which is basically a tRNA comes in and protein is being generated, and then they move to the next 3. So we profile the footprints of the ribosome and see that this actually occurs by 3. There's this periodicity. Basically, what your question asking is, the way I see it, is the question of how does a cell decide which RNA to turn into protein? And this is another side of investigation that we have in our lab. So the observation is this, according to your question, it is exactly this, that when we do ribosome profiling, we see that not all RNAs are made equal. which means, and then people in the field, they made this kind of term, which is called translation efficiency, which means some RNAs, they will make a lot of protein. So each RNA, there's a lot of protein being made from many molecules, many copies of the protein from one RNA. And some RNAs, they have low translation efficiency. So from this RNA, only a few of these proteins would be made. A few copies of the proteins are made. So, and then the question was, okay, is this important? And if it is important, what are the rules governing it? And this is also a question we like to ask in our lab. And why do we ask this question? Because we see this is not about cancer so much. It's more about, not about viruses so much. It's more about cancer. But we see in cancer that these mechanisms of select which RNAs to make into proteins are very important. And one of the cellular pathways which regulate this is called the mTOR pathway. So mTOR is protein which receives a lot of signals regarding the energetic state of the cell, and then activates or turns off or shuts off or turns on cellular processes which consume a lot of energy. So, for example, when we have a lot of energy and mTOR is active, it will signal to the ribosomes make a lot of protein. And when energy goes down, mTOR will signal to the ribosomes don't make a lot of protein. So this way, our cells, they use this regulatory mechanism in order to fine-tune how much protein they make. Now, what is interesting, when people use these techniques such as ribosome profiling and see what happens, what they found was that, again, not all RNAs are made equal. So when mTOR is signaling to, let's say, translation shut off, so don't make a lot of proteins, actually some RNAs are actually being translated more efficiently than they were before this shut-off signal. And then we see that the proteins which are encoded in these RNAs, they are proteins which are very important for the cell to deal with the stress. So this makes evolutionary sense.
Well, I was going to say, where do you think what's governing the translation? It's not just the attachment of the ribosomes, but internally in the ribosome itself. Is it complicated enough structure where it can still attach at a certain point, but then it still affects the translation, even though it's not just by location, but maybe internal action of the ribosome that also is another factor that governs translation or not?
For example, the ribosome is, let's say, 80% RNA and 20% protein. So the huge RNA structure with many proteins decorating it. When you look at different tissues or different biological conditions, you see the position of the ribosomes can change. So some ribosomes will have BCD attached to them, and some ribosomes will have protein BCDE attached to them. So these two ribosomes, if you give them a piece of RNA in a tube, they will synthesize the protein from this RNA. They're functional. But we think that perhaps the different compositions of ribosomes will allow it to translate more efficiently different sequences of RNA. This has not been proven, but this is an idea in the field, and people are asking the question. What we are more interested in is that we know a lot of the regulation of which RNA will be actually translated into proteins occurs at a step which is called initiation. So what happens is you have is born, let's say, and going to the cytosol, and now there are a bunch of protein binding to it. And their job is to prepare it so as the ribosomes can attach to them efficiently and translate. And when there is a signal, let's say that there's a lot of energy in the cell and everything is fine, then these proteins will attach to this and unwind, start it, and so on, and prepare it for the ribosome to work. When energy drops and mTOR is inactive, the composition of this complex, the newborn RNA, changes. And there are not these proteins there already to unwind the tangles and to allow the ribosomes to attach to it efficiently and be translated. So you can imagine a scenario where an RNA, which is more, let's say, tangled up, will be more dependent on the mechanism of unwinding than an RNA, which has a more, let's say, simple structure for the ribosome to recognize it and translate it. So perhaps under, for example, stress conditions, when there's scarcity of these unwinding proteins, the more complex RNA structured RNAs will be less efficiently translated. So this is something we're working on, because we think that the proteins regulating this transition from having the complex which unwinds everything to having it inhibited, I think is very important in cancer, and the proteins regulating this function are very important for the tumor cell to survive in the harsh metabolic conditions inside the tumor.
Very good. Well, Barak, what do you see as you figuring out in the next year or two? Anything getting close? Any hypotheses that you're testing and you're close to having a breakthrough on?
This idea of working on proteins which basically regulate which proteins are being made in the cell in cancer. So this is a work I'm doing with the collaboration with another lab in Germany, led by Gabriele Privier. He's a friend of mine. We did the postdoc together. And this idea we're very excited about, and we hope that it will happen very soon. Because what we found is a protein whose job is to inhibit translation or slow down translation. And we found that the cancer cells are very much dependent on it. And it's because you think about cancer cells, they want to grow very fast and they want to make a lot of proteins to allow this, to support this growth. And why would a protein whose job is to inhibit or to reduce protein synthesis would actually be important? So what we think it does is it allows the cells to down at times where there's glucose deprivation. So when the cell doesn't have enough glucose, and tumor cells, they love glucose. And if tumor cells, they don't have this protein, they cannot stop making fat and doing all the energy consuming things that they like to do when there's a lot of energy, and they keep on doing it even in times of starvation. And then they basically burn themselves out. So we're very excited by this, and hopefully it will be published soon.
Very good. Well, Barak, where can people find out your lab page? What's the address, and where can they see more of what you're doing?
You can Google Barak Rotblat, or you can, just Google Barak Rotblat Lab, and you'll find it. I'm on Twitter, so you can find me there.
So it's Barak, and then last name Rotblat. Very good. Well, Barak, thanks for coming on the podcast. I really appreciate it.
Thank you, Rich. It was a pleasure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.